Hello friends, welcome to Science Land. I'm Nikita and today we are going to talk about PHA production in plants. But before starting the video, I request you to subscribe to my channel. It really means a lot to me, and don't forget to hit the bell icon so that you get notified whenever I upload a new video. Thank you so much. Let's start. PHA production in plants, polyhydroxyalkanoates. Now, one of the most commonly occurring PHA is PHB, polyhydroxybutyrate, okay? And the video basically would be helping you to know the protocols or various studies which conducted the PHA production in plants. Now, the very first experiment was carried out in a plant called Arabidopsis thaliana. Now, people intended to produce PHB in the cytoplasm of this plant in the year 1990. Okay. Now, cytoplasm already has an enzyme called as beta-ketothiolase. Now, this enzyme is one of the enzymes which are required to produce PHB. There are total 3 enzymes. The first one is beta-ketothiolase. Second one is the acetoacetyl reductase, and the third one is PHB synthase. If you don't know, there is this video which I've made about PHB biosynthesis. It is there in the card. You can check the video out. Okay. So since the cytoplasm already has beta-ketothiolase, you only require the vectors for the other 2 enzymes. So the vectors were constructed, and the vectors were basically the binary vectors or the binary plasmids. Okay. Now, reductase is encoded by a gene called PHB gene, and synthase is encoded by PHB-C gene. Both the genes were incorporated into these plasmids, and they were driven under the cauliflower mosaic virus 35S promoter. Individually, they were transformed into the Arabidopsis plant. Now, why individually? Why can't you integrate both the genes in the same vector? Because plants cannot transcribe an operon with more than 1 gene. So, individually the plasmid was made. Now, once the individual plants were made, the cross was carried out, like cross-pollination, and the ultimate transgenic plant accumulated PHB. This plant produced 0.1% of dry weight of PHB in the leaves. Okay, so this was the experiment. Now, there are certain observations. 0.1% dry weight of PHB is very low. So, this low yield of PHB is due to certain reasons. What is the reason? The flux of the acetyl coenzyme A in the cytoplasm is not sufficient to produce PHB. So, acetyl coenzyme A is like a primary substrate to produce PHB molecule. It is there in the cytoplasm. But the amount it is present, it is very less. So, you will get less amount of PHB. The other observation was stunted growth. Now, since acetyl coenzyme A molecule is also used in other natural pathways of plant, like maybe plant hormone synthesis or sterol synthesis. So, the diversion of acetyl coenzyme A to produce PHB resulted in stunted growth. Okay, so this was the experiment, but there were certain takeaways from the study. Intention to produce PHB was actually targeted in the cytoplasm of the plant, but they also saw PHB production in nucleus and vacuoles. Okay, these both compartments also were found to produce PHB, but there was no PHB production in plastids and mitochondria. Maybe because they are double membrane structure, the PHB was not produced there. This was the major takeaway. Now, apart from this, the only unsatisfying thing was 0.1% dry weight. So, people would want to change this percent and they will improvise the protocol. And some of the protocols will be discussed now. Okay, so now let's talk about another protocol by Navrat, who tried to accumulate and produce a little more of PHB in plants. So he carried out this study in the year 1994. He suggested that plastids are a better alternative to produce the PHB. Now, why they are a better alternative? Because of the advantages the plastids provide. Like, plastids are double membrane structures, so the PHB which is produced is isolated, it would not leak out. Second, it naturally also stores starch molecules, lipid molecules, so it can easily store PHB. And the third one, it is a site of fatty acid biosynthesis. So the acetyl coenzyme A is present in good quantity and it will lead to PHB production quite easily. So these were the advantages. Now, plastids have one disadvantage, which is it is not stocked up with beta-ketothiolase enzyme. So he successfully produced PHB in plastid by creating 3 vectors. Okay, these 3 vectors are for 3 enzymes which are used for PHB biosynthesis: PHA-A, PHA-B, PHA-C. Okay, now all these binary vectors are from Agrobacterium tumefaciens PBI121. Okay, each of the 3 vectors had a chloroplast transit peptide from a rubisco small subunit isolated from P under the control of cauliflower mosaic virus 35S promoter. So basically what this means is the gene is there. Okay, 3 things are there in each of the vectors. The N terminal of each gene is fused to this transit peptide. What is this transit peptide? It's a signal peptide. It will signal the transportation from a particular place to another place. Okay, now this transit peptide here is isolated from P. Okay, and the entire vector is controlled by a promoter which is known as cauliflower mosaic virus 35S promoter. So 3 things: the gene itself, okay, the transit peptide and the promoter. 3 things in each of the vectors. Now each vectors are individually transformed into Arabidopsis thaliana plant, and each of these 3 plants are cross pollinated and it will give me a transgenic plant which produces PHB. The plant was observed to produce up to 14% of the dry weight of PHB. So, in the first protocol it was 0.1% of the dry weight and here it was 14% of the dry weight. It's a very big difference. Okay. Now, there were certain observations. No stunted growth was observed, but there was one disadvantage of this entire protocol. Leaf chlorosis was observed in plants which produced more than 3% of the dry weight of PHB. Leaf chlorosis as in the leaf is not able to produce chlorophyll sufficiently. That is known as leaf chlorosis. So, the initial problem of stunted growth is resolved, but another problem came up which is chlorosis. And further also, there was another person who improved this protocol. I'll talk about that protocol in a minute. Okay, now let's talk about another protocol which was given by Valentin and Bohemot, 1999 and 2000 respectively. What they did is they created a multiple gene transformation vector that allowed entire PHB genes to be inserted into one single locus in plant genome. Their idea was, the Naurats, the previous person's protocol, used 3 different vectors. Why can't we just clone the expression cassettes of all these 3 vectors into one and then use it for PHB production? They successfully did that. In fact, Valentin was able to produce 13% dry weight of PHB and Bohemot produced up to 40% of the dry weight of PHB in the Arabidopsis plant. But this high yield was accompanied by severe growth reduction and chlorosis. So that was, unfortunate, but more the yield, the growth reduction and chlorosis was more severe. Yet, these people didn't stop here. Now they started, coming up with protocols for another PHA, which was PHBV. If you don't know what is PHBV, the video is there in the card. You can check the biosynthesis of that. And they produced PHBV in Arabidopsis thaliana. So many such protocols come up, and they are actually a huge topic for research till date, and people will try to improvise the protocol. Now, I spoke only about Arabidopsis thaliana, but there are other plants as well, which are the host plants: tobacco, flax, cotton. All these are C3 plants which are actually used to produce PHA. C4 plants include maize, sugarcane. All these plants have been successfully shown to produce PHA. So the protocol which I mentioned in this video will be the same for most of these plants. There will be minor changes here and there. So that is the PHA production in plants. Thank you so much for watching. Bye bye.